So thanks everyone for joining us today for a cyber series that's going to talk about translating cannabis. And for me, I'm going to give really an overview of kind of where we came from, where we are now, and where we're going. And a large part of that, really, I have framed the title, and I'm going to say from the beginning that I'm going to be a little biased. I've framed the title being risks and benefits, but really, I'm going to really emphasize a lot about the risks today, and in the end, talk about some of the benefits in regard to therapeutic aspects of cannabis. And the reason that I'm raising more about the risk is I think today we're in this perfect storm. And I call it this perfect storm because of the changing sociopolitical landscape that has been surrounding cannabis for the past few years. And we started off with, earlier the government was like, but cannabis would make you go crazy and all of these things, and people started to distrust anything the government said about it. And then social media took over, and many people, promote how great cannabis is. But today we're in a perfect storm because there's increased information about cannabis, many of it incorrect on social media, but there's also increased THC concentrations in the cannabis plants that are consumed today, and even in the products that are bought in dispensaries, and there's a wide availability. Together with the fact that it's cheaper, there's a reduced risk perception, so a lot of younger people have access to it. So this time period that we're in is really a perfect storm for me, the developing brain, and the impact on cannabis. And so I'm going to talk a lot about that because one of the things that we know is that the neurodevelopmental processes, all from fetal, actually before fetal development, but all from fetal development into young adulthood, are impacted by the endocannabinoid system and by cannabis. And there are different behaviors that people have seen over the years in regard to exposure during fetal time periods. We're seeing now more and more children being exposed and the behavioral outcomes that we see, and young adults that has been studied for a very long time in terms of the effects of cannabis in the adolescent brain and the young adult brain. And so these are things that we know, and there's this long-term impact of developmental cannabis on the brain and behavior. So I'm going to start where I kind of started. For me, the question was, what is the impact, obviously, of cannabis on the brain, on the developing brain, and in studying human fetuses who, mid-gestational developmental period, we could see that the cannabinoid receptor, this is a gene expression, the cannabinoid receptor is where THC, the psychoactive component of cannabis, binds. And the cannabinoid receptor gene and protein are highly expressed in the brain, in the adult brain. They're actually the most abundant G protein coupled receptor in the adult brain. But during fetal development, it was interesting for us because it was predominantly expressed often in the more limbic regions, such as the ventral striatum, the amygdala, and the hippocampus. And a lot of research had started to show, and some of our work and collaborators showed that the endogenous cannabinoid system is really critical for hardwiring of the brain, because where the cannabinoid receptors are localized on these axonal tracts and growth cone, regulating neurodevelopment, and the endocannabinoid enzymes that synthesize and break down the endogenous endocannabinoid ligands. We'd even seen that in an unbiased strategy in an animal model that had prenatal THC, that a number of microtubule binding proteins related to cytoskeletal organization were altered with THC in our both our in vitro cell culture models and animals. But the thing that was important was that we could see this also in the human fetal brains in fetuses whose mothers had smoked cannabis. And specifically here, we could see SAPMIN2, which is also called SCG10, being reduced in the striatum and the hippocampus of these fetuses. We had also, like many people started off in a candidate gene type of strategy where we had looked at genes and proteins that are highly implicated in addiction. So for example, the dopamine D2 receptor, which we know in adult individuals with substance use disorders, reduced D2 is very characteristic of addiction, many different types of substances of abuse. And here we could see that the D2 receptor was already reduced in the stratum, particularly in the accumbens of fetuses whose mothers had smoked cannabis. We also saw that in the amygdala, significant alterations of the dopamine D2, and you could even from then in the prenatal period see even sex differences, where here was males that were more affected by their maternal cannabis use. So, you know, the main question for me has always been, you know, what are the long-term consequences and the specificity of some of that? And that's why we had focused a lot on our animal models, and we go back and forth. And we could see in our animal models that we could replicate a number of the changes that we saw in the human fetal brain. For example, here is dopamine D2. We see animals that were given, pregnant animals that were given THC, they have a reduction of the dopamine D2 in the accumbens, similar to the human fetuses. And we could see, importantly, that it lasted into adulthood. So that the changes that the impact from the prenatal THC is really long lasting. We studied a number of systems. I'm not going to go over all of them. I'll do some things a little bit later on, but just to say that it's, you know, a number of systems, as I said, the microtubule related genes and proteins, dopaminergic genes, GABA related genes, and epigenetic changes. And we also see that it miswired some of the cortical circuitry and even changed, you can see, the electrophysiological properties of these cells, where you can have impaired long-term depression. So all of this told us there are these significant neurobiological changes, but the question also, was there any impact on behavior? And for me, one of the big questions that we were looking at, both from a prenatal and an adolescent question, and driven in large part by the adolescent question, was is there this quote unquote gateway hypothesis, meaning that early exposure to cannabis, even during prenatal, could impact sensitivity to other drugs of abuse. And since opioids and cannabinoid receptors, they share a number of the intracellular signaling cascade, we focused initially on, is there a greater sensitivity, for example, for animals to self-administer heroin when they were adults, given only prenatal THC? And we could see these, again, with our earlier studies, that adult animals with prenatal THC, they self-administered more heroin, and even ran to the lever to get that first hit of heroin faster than normal animals. And if you gave them a mild stress, they started seeking the drug more. So we know that, like I said, this is where, a lot of, I've just summarized a lot of data, but it showed us that prenatal cannabis exposure could lead to protracted disruptions that you could see in adulthood on behavior, the neuronal morphology, electrophysiological properties, and gene expression, and a lot of it related to synaptic plasticity. So that's where we were. But a major question for me, and I think that for, I feel bad for my team, because we always try to have a translational component to it, to the research that we do. And so how do you study in humans this long-term effect of a prenatal exposure? And our group, we're masochistic, as I am, in looking at these long-term studies, but I think it's really important. And I then happened to meet a woman who was perhaps a little bit more masochistic than I am, Yoko Nomura, and was lucky enough to mentor Yoko during the first part of her K99. And Yoko really wanted to look at, and she'd been looking at prenatal stress in women, and developed a really great project, and we said, okay, let's collaborate together. We thought that this was, as I said, very masochistic to how do you study in utero exposures to different factors. And for Yoko, a major issue for her was stress. And unfortunately, we are living in a time today that is really very strong trauma and stress in our society. And however, in 2012, our city also experienced significant stress in the Superstorm Sandy hurricane. And I don't know if you guys can see that this is Manhattan underwater. Queens is cut off over here, and Queens was significantly impacted as well in many areas in the city. And so this was a stress, and in fact, a trauma that all of the women experienced. And so we also started looking more at aspects of, well, this is, so the phase one, the second, Yoko and her team, over 800 women were studied, and I've been following them and over time. And then we also started looking at cannabis, because definitely some of the women were smoking cannabis. And this has been a project I'll just talk a little bit about, just to give you some insights on how we try to translate some of this. So the study, there were placenta were collected, meconium, umbilical cord blood, and the kids have been followed up from they've been 6 months old in terms of physical development, neurobehavioral assessments, psychopathological measures, other also maternal psychological states, and so on. And the question for me in terms of, how do we get a sense about the environment in which the kids were developed? And so the placenta, a number of people call the third brain, because it's the link between what the mom experiences and what the fetus experiences. So it's that link between the fetal and maternal brain. And so we've been studying the placenta of these kids. And a postdoc, Greg Rampala, has been studying RNA sequencing data from these individuals. And one of the things that's clear is that, when you look at the RNA sequencing and you look at what are the genes that are changed, there are a lot of pregnancy-specific glycoproteins. So that was great that it wasn't saying we were studying the liver, but it's been fascinating what is changed. So first of all, most of the genes that are changed are regulated by the transcription factor NR3C1, which is the glucocorticoid receptor. And when you look at the genes that are most significantly changed, they relate to glucocorticoid biosynthesis, steroid biosynthesis, growth hormone receptor signaling, and the JAK cascade that's involved in growth hormone signaling. Indeed, many of the most significantly changed genes are essential for cortisol metabolism, progesterone synthesis, and as I said, the growth hormone, placenta-specific growth hormone. So what Greg then did, which is really important, was trying to figure out where these changes are. Is there any cell type specificity to where these changes are? And it was really fascinating, because the majority of the genes that were changed in relation to maternal stress was associated with this unique, well, it's not unique, it's the outermost layer of the placenta, which is and I always can never pronounce it, but the syncytiotropoblast. And these are unique in the context that they are really positioned at that maternal-fetal interface. So they form that epithelial barrier and are even critical for preventing maternal immune pathogens from getting into the fetus. So it's fascinating, as said, that the changes that we see in gene expression relates very specifically to this particular placental cell type. And moreover, the sandstorm's trauma stress was associated with a reduction in that gene expression signature, specifically within the cytotrophoblast cell types. So we know that, like I said, we saw that there are a lot of glucocorticoid signaling that was disrupted. And indeed, if you look at the genes that are within the syncytotrophoblast gene promoters, again, you see that it's the NR3C1 that is predominantly, which is increased in relation to sandy-storm stress, and that it's negatively correlated with the gene expression signature in that particular placental cell type. So it's extremely interesting that, as I said, that there's such specificity to prenatal exposure, or I should say maternal stress and intense stress. So this is not just any stress, it's this intense stress, really relevant, like I said, today's trauma, that you can see such specific changes in the placental transcriptome. So what about cannabis, since this is what, mainly said I would talk about today. And so cannabis exposure was also associated with differentially expressed genes in the placenta. What was changed, interestingly, were immune-related genes, and the genes were changed. And these included a lot of the pro-inflammatory cytokines and the enrichment you can see of these key immune function ontology genes, such as the interferon pathway, the cytokine-mediated signaling, and neurophil migration. When Greg looked again at the cell type specific signature, here you see that it's mainly related to, first there's an there's a suppression of the immune transcriptome in the placenta, and it's mainly predominantly enriched in monocytes. So those are the cells that are mainly, we see with a greater change with the maternal cannabis use for the placental transcriptome changes. And when Greg did gene co-expression analysis, again, indicating a significant immune functional organization of genes related to immune response, the immune system, defense response to viruses, and so on. So the prenatal cannabis definitely also alters the placental transcriptome, and we're still studying, a lot of the data, but one of the things for us that was interesting is that you can sometimes see this quote-unquote double hit consequence of prenatal cannabis and stress. And so, for example, here's a glucocorticoid receptor. Those that had both sandy storm stress exposure and cannabis exposure had the greatest change for the glucocorticoid receptor gene, while the opposite for the cannabinoid, the CNR1 gene. So we see these changes on gene expression profiles, but again, coming back to the question why we often go to our animal models is that, can you see an influence on future behavior? So what's the phenotype? And that's one of the critical things for this project. So Yoko and her team, they have been studying also, as I said, we've gone through in terms of a lot of the behaviors. So for example, just give us a couple of the behaviors that, some of the results for cannabis, at least specifically. So even from 6 months old, the kids show differences in their behavior. Here for the cannabis exposed kids, you see more distress and sadness. As they get older, you see anxiety. In fact, anxiety drive is a main drive that you see throughout their trajectory, throughout their development. But again, there's this synergistic interaction between cannabis and stress. So the greatest change of anxiety is in those who have both. You also see phobia and aggression in these kids. Importantly, we also see that there are changes in their hormone levels that are measured in the hair. So for example, cortisol levels are increased with cannabis exposure, as well as another adrenal hormone, DHEA. And when Greg looked, again, as I showed you before, that there was this reduction of the CNR1, the cannabinoid receptor, with cannabis, and there's a stronger effect with cannabis and stress. But it's also related, you can see, with some other, with the children's behavioral problems as they got older. For example, here we see depression and attention showing up in relation to the cannabinoid receptor. But if you look just globally at the transcriptome of the differentially expressed genes, yes, you still see that there's a significant correlation with their hormone levels with the placental gene expression patterns, but also you see that there are other behaviors that are also correlated to the placental transcriptome patterns. But perhaps the most fascinating thing for me, at least, is that when you look at the cytokine-mediated signaling pathway that is said was most significantly altered in relation to maternal cannabis use, you see that the differentially expressed genes, that there's a correlation of the genes with the cytokine, the genes that are enriched for the cytokine-mediated signaling, only in those who were only in those who were exposed to cannabis. You don't see the correlation of anxiety behavior in quote, I want to say normal, but in non-cannabis exposed in fetuses. So I don't want to say that again, because you see that in the kids, as they get older, anxiety is a prominent behavioral phenotype. And that behavioral phenotype is highly correlated to the cytokine-mediated signaling, but only those who were exposed to cannabis. We also see that there is a trend for an association with depression, but it didn't reach significance in relation to the cytokine network. So for this phase of our project, we're still analyzing a lot and trying to hopefully get set for the next phase as we continue studying these kids and how the placental transcriptome helps to give insights to their future behavior. But what is clear is that maternal cannabis exposure and definitely trauma alters the placental transcriptome, and you can see these protracted effects that correlate to the physiological measures, to behaviors, and a number of these behaviors are predictive of an early onset of psychiatric risk. And being able to study this population, we hope to see, one, what are things that can mitigate these potential risks, and two, in terms of does it indeed lead to this risk. But there's a huge correlation or interaction that we know between cannabis, stress, and the immune system that really is playing out here in these kids. So we can translate to some of the human work, and we're trying to now go back into our animal models to see whether or not we can replicate and so we can do more mechanistic studies. But we can now in our models, we used to look at the prenatal effects of THC into adulthood, and now we're trying to look earlier, since in our human project tells us that there are things happening very early. So, for example, Anissa, a postdoc in the group, has been looking at prenatal cannabinoid exposure from juvenile time periods. And for example, just quickly, you can see that even from very early in infancy, you see that there's ultrasonic vocalization disturbances in those with prenatal THC. Also, you see as they get older, there's changes in relation to social play. I'm not getting into all of the sex differences, but definitely there are sex differences both in our animals and in our in the human studies as well. They get into adulthood, you can see that, again, prenatal THC, here it enhances motivation for reward. So adult animals with prenatal THC, they will press a lever nearly over 100 times to get one chocolate pellet. That's probably me tomorrow night and Friday, but 100 times to get one chocolate pellet. So they're very sensitive to these positive rewards. But they also show something, like I say, similar to what at least we're seeing in our human population where there tends to be a depression, enhancement of depression-like phenotype. We've not studied anxiety as much in this model, and clearly that's something based on our human project that we're going to develop more. We also see that they show depression-like phenotype in this hedonic, this sucrose preference test, where if they're exposed once again to a stress, that enhances enhances their depression-like or their anhedonic-like state. So those are things, again, that here another stress later in life, which was the forced swim test stress, we can see these changes. So for us, the major question is, how do these gene expression and behavioral changes that we see later in life, how are they maintained across lifetime? And one of the things that we all know, and one of the reasons my group studies epigenetic mechanisms, is because we're looking at these long-term effects of developmental drug exposure, in particular cannabis. There are many different epigenetic mechanisms, but in order to try to understand, give us insights into what particular epigenetic mechanisms might come into play here, we initially did an unbiased strategy where we just looked at a lot of the genes that are critical for these different epigenetic processes, and these enzymes are involved in many of the epigenetic mechanisms. And what turned out in a number of the cohorts repeatedly was this particular gene, KMT2A. And KMT2A, which was also called mixed lineage leukemia MLL1, is a histone methyltransferase, and it's a mark of transcriptional activation. And it's known for a very long time that it plays a critical role in neurogenesis, in maintaining this transcriptional memory. And we could see that this gene, the overexpression of this gene, is there throughout the animal's lifetime, whenever we studied them, even though they were exposed to THC only during prenatal time period. You can see it on the gene expression, you can see it on the protein, like I said, at whatever age we have studied them. We can also go in and figure out whether or not KMT2A causally relates to some of the behaviors that we see. So for example, here we went in and knocked down KMT2A, and you can now normalize that increase of the motivated behavior, reward sensitivity that we saw earlier in these animals. So the whole goal was to be able to look at the KMT2A in terms of what is what is it doing in the genome? What are the genes where it's regulating? And unfortunately, the antibodies for looking at doing chromatin immunoprecipitation with chip assays didn't really work for us. And so we've tried an indirect strategy of looking at histone H3 lysine 4 H3K4 methyl transferase instead, or the H3K4 trimethylation as a proxy really for where KMT2A dysregulation might be targeting in the genome. And in doing that, Henrietta, working with another student earlier, and Randy Ellis now in doing a lot of the analysis, we then overlapped with RNA-Seq data to see, is there a particular region where trimethylation of lysine 4 may be impacting. And we see that in large part, what is changed relates to a lot about synaptic transmission and neuron projection guidance and so on. What then, when you look at, since trimethylation of lysine 4, H3K4, is an activation mark, we looked at the mRNA expression of those of genes that were downregulated. So if trimethylation of lysine 4 is up, well, okay, this one should be the opposite actually, sorry. But we were looking at both up and down genes, and again, no matter what you look, it's the synapse that comes in as being dysregulated. And this is for us in terms of trying to figure out more causally KMT2A in regulating these genes. Anissa has gone and developed CRISPR to go in and overexpress KMT2A to see, and this was done in collaboration with Prasant and Kristen, to really try to see, using another model here, progenitor cells, to see what also might be changed. And again, the overlap between prenatal THC and KMT2A specific changes, again, highlights the synapse. It highlights glutamatergic synapses, the postsynaptic density, as being significant for the changes that we're seeing on at least on a mechanistic level for this particular epigenetic regulation. So we right now are on we've been trying for a number of years in getting a better understanding of KMT2A, and I think that the CRISPR definitely now is a great strategy for us to use, because we do see that, as I said, prenatal cannabis exposure leads to this protracted disruption of epigenetic regulation, and in large part, like I said, we see KMT2A coming up, and that it's synaptic transmission in large part that we see is disrupted. So that's the prenatal part of the story. A huge part of the story for us is also about adolescent cannabis exposure, because that's another developmental period where there is significant dynamic neural processes ongoing in areas, for example, the prefrontal cortex, that's really critical for cannabis and a lot of psychiatric vulnerability. And we know, unfortunately, that cannabis use has gone up. In large part, we've taught people that cigarettes are bad and cigarette smoking had decreased, even though vaping, unfortunately, has increased in recent years. And so the perception that cannabis is not so bad is, as I said, one of the beginning, part of this perfect storm of greater use of high THC concentrations. And it's been well documented for many years that adolescent cannabis use is relevant to psychiatric vulnerabilities, such as increasing schizophrenia, increasing other outcomes, such as the use of other drugs, unfortunately suicide attempts. But those matters, the frequency matters, and the earlier you start matters. So again, we go to our animal models for trying to understand neurobiological consequences. And one of the things that we've seen repeatedly, and yes, there are nuances to this as well in terms of personality, even for rats matter, but in large part, adolescent THC exposure in adulthood increases their self-administration of heroin, for example. And one of the things that we know, as I just showed you from a lot of the human studies, that cognitive effects are prominent long-term with adolescent exposure. And here, Jackie Ferlin, postdoc in the group, has been looking at a number of the cognitive behavioral consequences of adolescent THC exposure. And for example, I'm just giving you guys a glimpse. Here's a rat gambling task, which basically is like the human gambling, Iowa gambling task, where there are like disadvantaged and advantageous choices for cards. Here, there's well, the rats don't have the cards, but it's a very similar model for the human in terms of looking at decision making in terms of cost benefit models. And one of the things that as I showed earlier, adult animals with adolescent THC exposure, they will self-administer more heroin. But we want to look at also what is the re-exposure if they see cannabis again and hear THC again later in life. And in animals adult animals that had adolescent THC, dose definitely matters. For here in cognition, it's those that have the higher dose of THC where they show greater impulsivity and when they are exposed to THC again. But a fascinating thing Jackie saw was that a few days after being re-exposed to THC, the animals that had the low dose, the animals that had the low dose THC were those that showed, and vehicles that actually showed decision-making impairments a few days later, and also greater impulsivity. So definitely high dose makes a difference in a long-term impact on cognition and in decision-making. But again, this shows that even low-dose exposure to THC still can increase risky choice. Jackie also looked at social interaction and social stress and could see that adolescent animals, or the exposure of to adolescents with THC in adulthood, they had a reduction in their social interaction, and it's specific because it's not for novel objects, it's specific to other rats. And then with social isolation, they increase their rewards sensitivity in self-administering chocolate, for example, so that the stress increased their social isolation stress increased their reward sensitivity. And it brings back an earlier things about stress. So here, measuring cortisol levels, 95 days later after their adolescent THC exposure, their cortisol levels in the high dose animals are still elevated. It's elevated usually closer in with the low dose, closer to their use, but here 95 days later. So stress is a huge aspect of the long-term effects of adolescent cannabis. And we know that the prefrontal cortex, as I mentioned earlier, is really critical for a lot of the cognitive processes that Jackie has studied, but importantly, the prefrontal cortex is the last region of the brain to gain full maturity. So in looking at that a number of years ago, here focusing, for example, on the prelimbic cortex, because that's one of the areas that undergoes perhaps one of the most pronounced developmental pruning during maturation of the prefrontal cortex. And it connects to a number of the brain regions that we feel are important for some of the behaviors that we've seen. And when we go in and look at these here, we took the pyramidal cells, for example, we didn't see anything in, for example, the length of the apical trees or basal trees of the pyramidal neurons. But what's changed was the complexity of these neurons. So with adult animals that had adolescent THC exposure, the morphology in terms of, like I said, the actual cortical branching differs. And a lot of this actually reminded us of earlier work that had shown with stress, once again, just emphasizing that some of the changes that we're seeing with cannabis may really relate to a stress activation. And here's an older study with previous students in the lab, Mike Miller, Ben Chadwick, where we said, okay, let's pick up those cells, and we needed to do it in a layer specific manner, since we had looked at the pyramidal cells and the gene expression, and really wanted to see what was happening in layer 2, 3 of these cells. And so these guys, they use laser capture and microdissection to pick up the cells, so that we could then do sequencing, and in doing the sequencing, we could then see that we could see that these animals there's a huge difference in the developing in the adult animals that have been exposed to adolescent THC exposure. I'm not going to go through all the details, but basically to say, excuse me, that there is a complete reorganization of the transcriptome in the placenta, in the pyramidal cells of these animals. So for example, there are only about 83 genes that are differentially changed in common in the normal development and the THC animals. And what has changed is really interesting. What's changed is, for example, it relates back again to this dendritic development, the cell projection morphogenesis, basically consistent with the morphological set of skeletal reorganization that we had seen in these cells. But importantly, there was a profound change in the epigenetic genes. So the histone modifications were significantly changed. And again, it was fascinating that what was changed, the genes that were changed, the strongest functional association for the epigenetic gene network came back to KMT2A and trimethylation of lysine 4. And as I said earlier, we know that KMT2A is a key regulator of morphogenesis, neurogenesis, and so on, but it has been hypothesized in schizophrenia-related and cortical dysfunction. So when Michael looked at the correlation between the genes that relate to dendritic regulation and epigenetic modifications, it was clear that there's a true reorganization. So here, for example, in fecal animals, there's not a great relationship between those genes. However, in adult animals that had adolescent THC, as you can see, there's a complete reorganization of this relationship between epigenetic modifications and dendritic regulation. Working with Panos Roussos, we also try to see what's the human relevance of these findings. And one of the things that was interesting was that a number of the gene networks that were changed in adults with adolescent THC were consistent with gene networks that were dysregulated in the prefrontal cortex of schizophrenia subjects. So the take home for most of this for us is that adolescent THC alters the structural development of prefrontal cortex that we can see morphologically, we can see genome-wide, and it relates to a reorganization of the epigenome, and a number of these gene networks relate to psychiatric risk. If there was, I'm going to quickly I just wanted to say that, we can see these early exposures, prenatal and adolescent exposure to THC and cannabis can have a long-lasting effect across a lifespan. But there's also the question of subsequent generations. And again, because I'm masochistic and apparently attract masochistic people, the question many years ago we started to ask was, what's the impact on subsequent generations? And just want to show one quick thing, because is there a germline effect? When we started studying that question, many people, it was very tough again to get things published because people didn't believe that there could be these germline effects that could go across generations. But we did see that, if you have adolescent animals given THC in a similar way that we have for our normal paradigms, and then had other animals raise their offspring and studied their offspring, Henrietta was able to see behavioral differences, glutamatergic changes, so electrophysiological changes. But I just point out one thing. Could also see that the gene expression network that is significantly changed in the striatum of these animals that were not exposed directly to THC themselves, it was their parents, when their parents were teenagers. Again, it comes back to KMT2A and histone, trimethylation of histone 3, or lysine 4 and histone 3. So we're still trying to figure out the significant role of KMT2A, and Greg Rampala has some really beautiful mechanistic studies of how germline transmission can occur. But this has been, a fascinating thing that this epigenetic mechanism keeps coming up with a lot of developmental cannabis. So I did promise to talk about benefits as well, so risk benefits, and since there is a little time, I wanted to just add a few things that we've seen regarding to cannabis of benefits. So, I mean, one of the things that's critical to understand, and I think most people do now, is that the cannabis plant is extremely, it's a complex plant. It has over 500 chemicals, and most of these are, over 140 of them are cannabinoids. And these cannabinoids, we mainly focus on THC because that's the psychoactive cannabinoid that leads to the reward, the high. That's why people, smoke cannabis recreationally. But a number of the other cannabinoids also have biological activity. And one in particular, cannabidiol, you know, in recent years has gotten a lot of attention. We had studied it before that, but one of the things that we could see was that in our animal model, CBD is actually very different to THC. So as I showed you before, giving animals THC, they will, for example, increase their heroin self-administration behavior. But when we gave CBD, we actually saw a different pattern. It decreased heroin-seeking behavior. So when animals are learning to self-administer a drug, cues in the environment are associated with that for them to obtain the drug. So for example, a light goes on in the environment, and after a while they associate the light with getting the drug. So just showing them the light alone, they start pressing the lever. And this is why this shows that CBD reduced this environmentally induced heroin-seeking behavior. And importantly, the effects of CBD lasted weeks after their final administration of CBD. And we know that from a lot of our work in our postmortem human studies of heroin addiction, that there's a significant reorganization of the glutamatergic system there as well, and changes in synaptic plasticity, which again is characteristic of a lot of addiction. And when we looked at CBD effects on, for example, glutamatergic genes, so this is a prototypical GLUA1 gene, we could see that animals that self-administered heroin had impaired AMPA receptor, and CBD normalized it. We also know that heroin disrupts the endocannabinoid system in part, and here's CNR1, the gene, and the CB1 receptor, and CBD normalized the upregulation that heroin normally caused with that. So we started to develop a program to look at cannabidiol as a potential treatment for opioid relapse. And we focused on craving because just like the animal model, a lot of environmental cues for humans who suffer from substance use disorders are really the things that trigger their relapse, triggers their craving. And so it's environmental cues, it's stress, and it's obviously exposure to the drug again. And so we did a first pilot study, randomized, double-blinded control, and we could see if we showed people a cue with a heroin cue and they had received placebo, they craved. But we could also see that they actually increased their anxiety, and CBD reduced that. And like the animals, we saw when we brought them back a week later, it still reduced their general craving. But that study was very small, so we, for a number of reasons. So we carried out another study, and here at Solstice, who's really great, and a former clinical coordinator, Sharon Spriggs, we again looked at the same things and looked at craving. So similar to what we saw before, it replicated. So if you those people with a heroin use disorder, if they are shown a heroin cue, and got placebo, they craved. But if they were given CBD, it reduced their craving. And it lasted a week later when you brought them into the lab. Similarly, with anxiety, again, heroin induces anxiety tremendously, and CBD reduces that. A week later, CBD still had a week after its last CBD administration, it was still reducing their anxiety. This time around, we could measure, we carried out physiological measures, and here looking at stress hormone, cortisol levels, and CBD, if they're given placebo, their cortisol levels go up, and CBD reduces that. And their heart rate, similarly, they see a drug cue, their heart rate goes up, and CBD reduces that. So we do know that CBD holds some promise for opiate use disorders, and some other addiction disorders are now being studied, and in particular also aspects of anxiety and craving. But there's a long way to go. We need to know the doses, the formulations, you know, a lot of things that we still don't know about CBD. And we were lucky to get two grants, two clinical studies are now being conducted in a large clinical trial with CBD in heroin use disorders, and another with neuroimaging to look at CBD to see its effects directly in the brain. So as we said, you know, the risks and benefits for cannabis are very profound. because many people think that now, cannabis cures everything. But unfortunately, it's not cannabis. It's specific cannabinoids. So we see more of risk in regard to substance use disorders with THC, psychosis with THC, promoting anxiety with THC, and the impact that I've showed you today on the developing brain, that a lot of this relates to THC. And in contrast, the benefits that are starting to emerge are a lot dealing, for example, with CBD for different substance use disorders, opioids, even tobacco, cannabis, and alcohol. Also, psychosis. A number of studies have shown CBD's beneficial effects with psychosis, and as I showed with anxiety, not only in people with a substance use disorder, but without a substance use disorder, CBD's impact on anxiety is now being replicated. There's still a lot of controversy about CBD and developmental disorders, and for some developmental disorders, there may be needed some THC depending on the particular disorder. And obviously, childhood epilepsy, CBD has been, it's the only FDA-approved indication for cannabis or CBD today. And a lot of studies are being done for pain and inflammation, where CBD definitely will play a role, but THC is also being potentially needed for certain types of pain. So with that said, I've covered a lot of ground showing you some of our developmental cannabis work, and a large part of it, this top line here with Henry Desutzer, really leading the way with a lot of our masochistic studies in the transgenerational effects that she was joined with Greg, and Greg doing an amazing job. Also with our human fetal project, Anissa with the prenatal, and Jackie with the adolescents, and Ren helping a lot with all our computational things as well. I do want to give obviously a shout out to the whole team. We do a lot of long-term behavioral studies, and Joe, James, Annie are phenomenal in maintaining our behavioral program, and we couldn't do anything without them. So really appreciate all the hard, long hours everybody puts in for these studies. And Salsis, I didn't go through much with all of the clinical studies today, but it's been a great clinical partner for our program there. But as I said earlier, a huge shout out to Yoko Nomura and her team, Lexi, Patricia, and Melissa. It is really challenging to do human longitudinal studies, much less starting from the prenatal period. And we now have actually a HEAL project. I have a HEAL project with Verley Berging for the opioid exposure, and trying to study longitudinally kids, and we'll see how those go, but it's really challenging. So Yoko, thank you so much for partnering on this ride. So I'll take any questions that are there.
Yasmin, 5 milligrams of THC is equivalent to what?
Oh, wait 1 second. I just realized one thing, but sorry. And I forgot my big shout out to my clinical team who are carrying out all of the CBD clinical studies. So that's the group there that has been amazing with our human clinical work. Sorry. 5 milligrams. Say the question again.
5 milligrams of THC is equivalent to what in real life?
So 1.5 milligrams for THC, our classic amount that we used to use, was equal to about a joint. And then the THC concentration started going up. So that's why the 5 milligrams now is more consistent to what's on the street today.
Thank you.
Hi, Yasmin. You have a few questions in the chat box. I don't know if you can see that.
Oh, okay. Let me try to get to that. Can you say a bit more about the pregnant mothers and the cannabis use versus not? Were they matched in education, IQ, psychiatric? Thanks, Erin, for that question. And yes, so, I mean, they're not matched for these things because this is the reality of women who were pregnant. But they didn't differ when we looked at a number of their, for example, education and so on. they differed in some anxiety and depression for the moms. For a lot of the statistics, we controlled for a lot of the demographics and so on. And definitely that's why we have our animal models to try to dissociate or to, the confounds that are there with human studies. But the human studies are critical because I don't think that we would be looking at the placenta if we hadn't seen the human studies, but now we're starting to look at the placenta. So I think the human studies are really important, and the immune system to guide us. So thanks. Another person says, great talk, thank you. I'm surprised that a single exposure to THC in adolescents would result in an amp up HPA. So I didn't give a detailed overview of the study design. It wasn't a single injection of THC. THC was injected like every third day across adolescents to replicate more of a more recreational type of periodic cannabis use than just, say kids just tone down their minds. So it's not a single exposure. The thing that's fascinating is that in adulthood, when they're given just one re-exposure to THC, that it does have this long-lasting effect. And so I think that people don't, even in our human subjects, we do see that the court is changed. Their HPA access is changed. Fiona, are effects seen in offspring germline per se, or does growing up in a stressed environment leads to the same epigenetic changes? That's a great question. And for me, it's a combination. Clearly, there's something happening germline that we're now, as I said, from Greg's work, we're able to see some of the mechanisms there that can lead to that. But the environment that you're also raised in makes a difference. So for our human study, that's the challenge, right? Because they have the prenatal exposure, but they're still being raised in environments perhaps that are, for some that may have greater stress than others. And it's that combination. So to me, it's the combination of both the environmental stress and the germline impact as well. And, I do think an aspect of the results that we've gotten that I do think is quote unquote a plus is that many of the changes are mediated are epigenetically mediated, which means that they could be reversed. They can be reversed by perhaps behavioral interventions. So the fact that we see such epigenetic dysregulation, I think it's actually a good thing because it does give hope that we can reverse some of these behaviors. Another question, will you be doing a similar study following COVID-19? That is what I think I definitely won't, because I've now hit my masochistic wall. Yoko does want to, but we're having challenges in terms of, as I said, these studies are very long term, and I think that they're very important because it does show that what pregnant women, the trauma of just that big trauma, the impact that it has on placental transcriptome, that impacts on their offspring. And so, it really, I think, is critical to do the COVID. Sinai has started a project and they're following up, and Verdi Berging is one of the people part of that. And so we'll see in this COVID-19 moms some of the outcomes there, but I don't think I'll join as closely. What is the reason for such different effects between THC and CBD, since while the chemical structures are so similar? So they do bind to different receptors. Even the chemical structures are similar, but the differences are clearly enough. So for example, while THC binds to the CB1 receptor, it's an agonist, it's a partial agonist of the CB1 receptor. CBD is more of an inverse agonist. And there are many other systems that CBD, the mechanism of action, let's put it this way, for CBD is still being explored, but it definitely has a very different profile than THC in the receptor binding activity. And another question that's very interesting thing, just to clarify, if mothers refrain from cannabis use during pregnancy but were users in the past, does this impact the child? So that's part of what our our multi-generational studies is trying to help us get insights on. To be honest, we have mainly studied the male line, and we're doing human studies with sperm from men in terms of, again, translating what we've seen in our animal models. So I do think that there is a potential to impact on the child based on our animal models. But again, the one thing about the animal models is that it tells us the potential of what may be possible. But humans, we have a much more complex life. And coming back to the question someone asked earlier about the environment can help to change some of those things from cannabis use in the past.
Thanks again, Yasmin. Hope everyone is safe and well, and look forward to the next seminar in the series.
Thank you all. Thanks so much.